Hello students, welcome to EPG Pathsala. I am Dr. R. Papamal from Department of Science and Humanities, Faculty of Engineering, Avinashlingam Institute for Home Science and Higher Education for Women, Coimbatore. Now we are going to see about sources and types of free radicals and oxidative stress. Free radicals are the products of normal cellular metabolism. A free radical can be defined as an atom or a molecule containing one or more unpaired electrons in its outermost orbital and is capable of independent existence. The free radicals can be produced from either endogenous or exogenous sources. Majority of the free radicals are produced from oxygen atoms or called reactive oxygen species such as superoxide ion, hydroxyl radical, hydrogen peroxide and singlet oxygen. Oxidative stress occurs when the production of reactive oxygen is greater than the body's ability to detoxify the reactive intermediates. This imbalance leads to oxidative damage to biomolecules and genes within the body. The body naturally produces antioxidants like superoxide dismutase, catalase and peroxidase enzymes as a means of defending itself against free radical. The objective of the module is to understand the different sources and the types of free radicals generated in the body, to know about the formation of oxidative stress in our body. Free radicals. The unpaired electron of a free radical makes it unstable, short-lived and highly reactive. Because of their high reactivity, they can attack electrons from other compounds to attain stability. Thus, the attacked molecule loses its electron and becomes a free radical itself, beginning a chain reaction, finally damages the living cell. The free radical is generally denoted by a superscript dot. Both reactive oxygen species and reactive nitrogen species are collectively form the free radicals and other non-radical reactive species. These are the sources of free radicals: endogenous sources and exogenous sources. The primary source is from our body during the energy production process within the cells. 98% of the oxygen we breathe is used within our cells in the production of energy. The result of an energy producing activity is that a very small amount of oxygen that was not used loses electrons, creating free radicals. Another important source which creates a lot of free radicals is the presence of toxic heavy metals in the body, especially those caught up in the cellular tissues. All transition metals, with the exception of copper, contains one electron in the outermost cell and can be considered as a free radical. The metals which are the major free radicals creating agents in the body are mercury, calcium, aluminium, lead, chlorine, iron and cadmium. From foods that we consume that may have a destructive potential. An example would be fried foods in high temperature, which causes the bonds in the molecule to become unstable. During normal metabolism of fat, carbohydrates and proteins. And the stress, especially the oxidative stress, all chemicals and some endocrine hormones create free radicals. And low blood supply generally occurs during the heart attacks and strokes forms free radical. The endogenous sources of free radicals such as reactive oxygen species include different cellular organelles such as mitochondria, peroxisome and endoplasmic reticulum where the oxygen consumption is high. Coming to the mitochondria, the superoxide anion is converted to hydrogen peroxide by the action of mitochondrial superoxide dismutase. Hydrogen peroxide can be detoxified by the catalase and glutathione peroxidase. The other mitochondrial components which contribute to the formation of reactive oxygen species include monoamine oxidase, alpha-ketoglutarate dehydrogenase and glycerol phosphate dehydrogenase. Coming to the peroxisomes, In the case of peroxisomes, the respiratory pathway involves the transfer of electrons from various metabolites to the oxygen, leads to hydrogen peroxide formation, but is not coupled to oxidative phosphorylation to produce ATP. Instead, free energy is released in the form of heat. The other free radicals produced in peroxisomes include hydrogen peroxide, superoxide anion, hydroxyl radical, and nitric oxide. Coming to the endoplasmic reticulum, the enzymes of endoplasmic reticulum such as cytochromes P450 and diamine oxidase contribute to the formation of reactive oxygen species. Free radicals are also created externally in the following situations. The first one is environmental contaminants such as pollution. Cigarette smoke in particularly increase the free radical load abundantly. The second one is ionizing and ultraviolet radiation such as from overexposure of the sunlight and prolonged exposure to X-rays. Third one is over-exercising. The fourth one is toxic chemicals and pesticides. Fifth one, petroleum-based products. Sixth one, unhealthy foods such as processed foods and fried foods. The seventh one is toxic products found in furniture polish and paints. Example, toluene, benzene and formaldehyde. So these are all the free radical generating substances like fried foods, alcohol, tobacco smoke, pesticides and air pollutants. These are all exogenous sources. Now we are going to see about the UV rays. Depletion of stratospheric ozone makes the upper atmosphere more transparent to ultraviolet light. Skin is the most susceptible target organ to UV radiation. Exposure of skin to UV ray that can lead to a premature aging and carcinogenesis. There is a marked evidence suggesting that UV light induced damage in the skin is because of free radical generation. Coming to the atmospheric pollution point of view, environmental pollutants range from metals, halogenated compounds and air pollutants damage the cellular macromolecule in the body due to the absence of or lack of antioxidants. Coming to the arsenic point of view, the soil, water and air contain a metal contaminant such as inorganic arsenic. It has been readily absorbed by the human beings and induce the formation of tumors in the bladder, prostate, skin and liver. Inorganic arsenic undergoes series of methylation steps by using S-adenosyl L-methionine as a methyl group donor, and methylated form of arsenic act as a carcinogen in vivo. The next compound is a haloalkanes. They are formed from alkanes with chlorine and have been used in industries as a solvents and antiseptics. Some of the haloalkanes are present as contaminants in drinking water and also in chlorinated swimming pool. They are highly hepatotoxic and also have a carcinogenic property in humans. The next compound is a hydrazine. Hydrazines are environmental pollutants and are induce liver toxicity, carcinogenicity and mutagenicity. Hydrazine derivatives produce free radicals upon oxidation, causing DNA oxidation and damage. The next pollutant is a smog. It is usually appear during hot sunny weather, where it appears as a brownish haze that can irritate the eyes and the lungs. Gases present in smog are nitric oxide, sulfur dioxide and ozone and oxidants which induce organ and cellular damage by generating free radical species. Nitric oxide rapidly reacts with the molecular oxygen to generate highly reactive peroxynitrite, which is known to oxidize DNA, proteins and lipids. Sulfur dioxide reacts with water to produce hydrated sulfur dioxide, which has been shown to generate sulfite and sulfate anion radicals catalyzed by human myeloperoxidase. Sulfite is also to be used as a food preservative. Exposure to sulfite has been reported to induce allergenic reaction and bronchoconstriction in sensitive human populations. It is now believed that free radical formation from sulfite may be involved in damaging sensitive proteins. Next factor is stress. The pressures common in industrial societies can trigger the body's stress response. In turn, the stress response generates free radicals in tremendously. The stress response raises the body's energy creating apparatus, increasing the number of free radicals as a toxic byproduct. Moreover, the hormones that mediate the stress reaction in the body is cortisol and catecholamines will themselves generate into a particularly destructive free radicals. Researchers now know one way in which the stress may cause diseases. A stressful life can produce mass of free radicals. The next factor is dietary habits. Dietary deficiency of selected antioxidants, micronutrients and their association with known risk factor in the pathogenesis, occurrence of various cancers. Concurrent correction of plasma antioxidant level may be significant part of optimal nutrition help to prevent early stages of cancer. High fiber diets which will prevent the cancer. Scientists postulated that microorganisms associated with low dietary fiber favor the production of cancer and cancer producing substances. If we include diet which is rich in antioxidants in our daily food intake rather than pills containing nutrients may reduce a person's risk for developing a cancer. Now we are going to see about the types of free radicals. These are all the types of free radical. Oxygen-centered radical, sulfur-centered radical, carbon-centered radical and nitrogen-centered radicals. The reactive oxygen species and reactive nitrogen species play a 70% job as both beneficial and toxic compounds to the living system. At low level of reactive oxygen species and reactive nitrogen species have a beneficial effects and involve in various physiological functions such as defense against pathogenic microorganisms and in a number of cellular signaling pathways. But at a higher concentration, both reactive oxygen species and reactive nitrogen species generate oxidative stress and nitrosative stress respectively, causing potential damage to the biomolecules. The oxidative stress and nitrosative stress are developed when there is an excess production of reactive oxygen species and reactive nitrogen species on one side and a deficiency of enzymatic and non-enzymatic antioxidants on the other side. The excess of reactive oxygen species can damage the integrity of the various biomolecules including lipids, proteins and DNA, leading to increased oxidative stress in various human diseases such as cardiovascular disease, respiratory disease, diabetes mellitus, neurodegenerative diseases and rheumatoid arthritis, cataracts and aging process. These are all some of the reactive oxygen species and reactive nitrogen species. In both the cases, we are having radicals and non-radicals. Under that reactive oxygen species radicals, superoxide, hydroxyl, alkoxyl radical and peroxyl radical will come. Under that non-radicals, hydrogen peroxide, singlet oxygen, ozone, organic peroxide, hypochlorous acid, hypobromous acid will come. And another thing is reactive nitrogen species. Some of the radicals are nitric oxide and nitrogen dioxide. Non-radicals are peroxynitrite, nitrosyl cation, hydroxyl anion, and dinitrogen trioxide, dinitrogen tetraoxide, nitrous oxide, and peroxynitrous acid, nitrile chloride. So these are all some of the species. Coming to that reactive oxygen species radical. The first one is we are going to see about superoxide anion. Superoxide anion radical is the most important widespread reactive oxygen species formed by the enzymatic process of autooxidation reaction and by non-enzymatic electron transfer reaction in which an electron is transferred to a molecular oxygen. It is mostly produced within the mitochondria and its reactivity with the biomolecules is low. Superoxide radical react with another superoxide radical in a dismutation reaction in which one radical is oxidized to oxygen and other is reduced to hydrogen peroxide. So this is the equation. Two superoxide anion radical combined together, we are getting that hydrogen peroxide and oxygen molecule. The next ion is radical is hydroxyl radical. Hydroxyl radical is a neutral form of hydroxide ion and is a highly reactive free radical. It can strongly react with both organic and inorganic molecules including DNA, proteins, lipids, carbohydrate and cause severe damage to the cell. The next one is peroxyl radical. It is derived from oxygen in living beings. The simplest form of peroxyl radical is a peroxyl radical which is formed by the protonation of superoxide. It starts the fatty acid peroxidation and also can promote the formation of tumor. The next reactive oxygen species comes under the non-radical category. The first one is a hydrogen peroxide. It is formed in living systems by a dismutation reaction catalyzed by the enzyme superoxide dismutase. The major antioxidant enzymes that can eliminate hydrogen peroxide include catalase and glutathione peroxidase. Singlet oxygen. It is an electronically high excited metastable state of molecular oxygen and is a highly reactive, toxic reactive oxygen species. It is a highly potent oxidizing agent that can cause DNA damage and tissue damage, which in turn leads to occurrence of various diseases. The next one is ozone. It is a powerful oxidant. It may be produced in the living system by water oxidation pathway, which plays an important role in inflammation. It can also lead to chromosomal aberrations. It may be due to the direct attack by ozone or by the free radicals which is generated. The next one is a reactive nitrogen species. Coming to the radicals point of view, the first radical is nitric oxide. It is a small molecule generated in tissues by different nitric oxide synthases, which convert L-arginine to L-citrulline. Nitric oxide play multiple role in which it can also act as an important cellular redox regulator and to regulate enzymatic activity by nitrosylating the proteins. The next one is a peroxynitrite. Peroxynitrite is formed by the reaction between superoxide anion and nitric oxide. It is highly toxic and can directly react with the carbon dioxide to form other highly reactive nitrogen species radicals or reactive nitrogen species non-radicals. Targets of free radicals. When there is an imbalance between the free radical production and the antioxidant defense, the free radicals will be produced in higher concentration, leading to oxidative stress and nitrosative stress. Since these free radicals are highly reactive, they can damage all the three important classes of biomolecules such as nucleic acid, proteins and lipids. First, we are going to see about the DNA, deoxyribonucleic acid. Both reactive oxygen species and reactive nitrogen species can oxidatively damage the nucleic acid. The mitochondrial DNA is more prone to that reactive oxygen species attack than the nuclear DNA because it is located in the close proximity to the reactive oxygen species generated in the place. Reactive oxygen species, most importantly the hydroxyl radical, directly react with all components of DNA such as purine and pyrimidine bases. Deoxyribose sugar backbone and causes a number of alternations including single and double stranded breaks in the DNA. Finally, the mutagenic DNA lesions will be formed and is responsible for the carcinogenesis. The next nucleic acid is a ribonucleic acid. The reactive oxygen species can attack different RNAs produced in the body. The RNA is more prone to oxidative damage than DNA due to its single-stranded nature and a lack of an active repair mechanism for oxidized RNA, less protection by proteins than DNA, and moreover these cytoplasmic RNAs are located in close association to mitochondria where the majority of the reactive oxygen species are produced. RNA damage product and its levels are raised in various pathological conditions like Alzheimer's disease, Parkinson's disease and atherosclerosis. The next biomolecule is proteins. The protein oxidation can be induced by radical species such as superoxide anion, hydroxyl radical, peroxyl, alkoxyl, hydroperoxyl, as well as the non-radical species such as hydrogen peroxide, ozone, hypochlorous acid, singlet oxygen and peroxynitrite. The sulfur-containing amino acids such as methionine and cysteine are more prone to oxidation by reactive oxygen species and are converted to disulfides and methionine sulfoxide respectively. The reactive oxygen species induced oxidative damage of amino acid residues such as lysine, proline, threonine and arginine yields carbonyl derivatives. The presence of carbonyl group in proteins has been considered as the marker of reactive oxygen species mediated protein oxidation. An increase in the level of protein carbonyl is observed in a number of pathological conditions such as Alzheimer's disease, Parkinson's disease, muscular dystrophy, rheumatoid arthritis, diabetes, atherosclerosis and aging process. The next biomolecule affected by the free radicals are lipids. The membrane lipids, especially the polyunsaturated fatty acid residues of phospholipids, are more prone to oxidation by the free radicals. The lipid peroxidation is very important in living systems because of its involvement in various pathological conditions. Now we are going to see about the oxidative stress. Oxidative stress is used to describe the condition of oxidative damage resulting when the critical balance between free radical formation and antioxidant defense is unfavorable. Oxidative stress arising as a result of an imbalance between free radical formation and antioxidant defense is association with damage to a wide range of molecular species which includes lipids, proteins and nucleic acids. Short-term oxidative stress may occur in tissues injured by trauma, infection, heart injury, toxins and excessive exercise. These injured tissues produce increased radical generating enzymes such as xanthine oxidase, lipoxygenase and cyclooxygenase. Finally, leads to the activation of phagocytes, release of free iron and copper ions, or a disruption of the electron transport chains of oxidative phosphorylation, producing excess of reactive oxygen species. The initiation, promotion and progression of cancer as well as the side effects of the radiation and chemotherapy have been linked to the imbalance between the reactive oxygen species and the antioxidant defense system. Reactive oxygen species have been involved in the induction and complications of diabetes mellitus, age-related diseases and neurodegenerative diseases such as Parkinson's disease. Here we are seeing about the oxidative stress and that superoxide dismutase, that is an antioxidative enzyme, how the balance is maintained. If the balance is not maintained means there is prone to get a cancer formation. Human diseases and oxidative stress. Free radicals are involved in many pathological conditions such as many types of diabetes, neurodegenerative diseases, cardiovascular diseases, cancer, cataracts, asthma and rheumatoid arthritis inflammation. The first one we are going to see about the diabetes mellitus. It is a heterogeneous group of chronic disorders characterized by elevated blood glucose level is called hyperglycemia due to defective insulin secretion in the case of type 1 diabetes and resistant to insulin action in the case of type 2 diabetes. Diabetes mellitus is associated with the increased production of free radicals or decrease the activity of the antioxidant systems, which leads to the formation of oxidative stress. Both the mitochondrial and non-mitochondrial derived reactive oxygen species contribute to the oxidative stress during the diabetes mellitus. The next disease is neurodegenerative diseases. The central nervous system is especially prone to the oxidants due to the presence of high lipid content, high consumption of oxygen and low levels of antioxidant enzyme. The oxidative stress state has been also associated in several neurodegenerative diseases such as Alzheimer's, Parkinson's and multiple sclerosis. The next disease associated with the free radical is cancer. It is one of the leading causes of death in humans. Free radicals cause different types of chemical changes in the DNA. Thus, they could be mutagenic and involved in the causes of different types of cancer. The epidemiological studies postulated that low levels of antioxidants are associated with increased risk of cancer. The next disease is atherosclerosis. It is a pathological condition commonly referred to as hardening of the arteries. Hyperlipidemia is a major risk factor for the formation of atherosclerosis. A profound imbalance of the oxidants and antioxidant resulting in the oxidative stress is observed in atherosclerosis. The next one is hypertension. Persons with hypertension are at an increased risk for stroke, heart disease, kidney failure. Free radical induced oxidative stress in part contributes to endothelial disinfection and the development of hypertension. Increased reactive oxygen species generation eliminates nitric oxide radical by forming peroxynitrile radical anion and thus reducing the nitric oxide radical. Bioavailability, which leads to decreased endothelium dependent vasodilation, leads to hypertension. Another disease which is related with the free radical is cataract. It is characterized by the opacity of the eye lens and decreases the amount of incoming light and resulting in visual impairment. The free radical induced oxidative stress is considered as one of the major risk factor leads to cataract. And another disease which is related with the free radical problem is rheumatoid arthritis. Rheumatoid arthritis is a chronic disease which affects approximately 2% of the total world population, and women are affected more than men. Particulate and gaseous phases of the smoke contain high concentration of free radicals that can interact with the DNA and could cause genetic changes, and its activation responsible for the development of rheumatoid arthritis. And another disease which is related with the free radical problem is asthma. Asthma is the most common disorders in the airways of the lungs, and it is one of the major global health problems. Numerous studies have suggested that oxidative stress is formed by excess production of various free radicals or by an insufficient antioxidant defense system in asthma, and thus it contributes to the tissue damage which is induced by inflammatory cells. Coming to the conclusion part of this module, free radicals are very harmful, but their production within the body in limited amount is an essential players in the immune system. But at higher concentration, reactive oxygen species generate oxidative stress and damage the biomolecules including lipids, proteins and DNA, leading to various human diseases such as respiratory diseases, diabetes mellitus, neurodegenerative diseases, rheumatoid arthritis, cataract and aging process. Consumption of food that contain antioxidants rather than pills, we can able to avoid the free radical related diseases, especially the cancer, Alzheimer's and all. Thank you.